Right, this is part C, the third and final part of lecture 3 on bacterial and archaeal cell structure, the second lecture of a two-part series. This portion will focus on understanding the structure and function of endospores. Endospores are specialized or differentiated cells. They're different from the vegetative cells that members of the species that make endospores can also make. Endospores are highly resistant to heat, harsh chemicals, radiation, nutrient depletion, and desiccation or dryness. They therefore function as survival structures, enabling the microbe to persist in a dormant, non-metabolically active state, ready to regenerate or sporulate once conditions improve. They're made by particular genera of gram-positive bacteria. Really common or well-studied genera that make of bacteria that make endospores are the Bacillus and the Clostridium, as well as several others. I'll give you some examples of specific bacteria at the end of this portion of the lecture. So this is a view of an endospore as taken using either fluorescence microscopy over on the right or to the left. This position here, this would be done with an electron microscope using electron microscopy. There is a protein exterior that includes the exosporium, spore coat, and core wall. All of those structures are protein-based, and you can see that they're very, very thick. So it's a very rigid, highly protective structure. The cortex is made out of the molecule that we discussed in class earlier, peptidoglycan. Okay, so by the way, endospores are something that are in fact unique to the domain bacteria, just as the molecule peptidoglycan is unique to the domain bacteria. So there's a very thick layer of peptidoglycan, also adding a protective function to these structures. The interior, or the core, is bounded by, a cytoplasmic membrane. So that would run just in here, all right? And it contains very dehydrated cytoplasm. It's a very low water content in endospores. It has ribosomes within it, and of course, there's a chromosome there. So you have to have DNA. Remember, the whole purpose of these structures is survival, so you have to have all of the cell machinery required to resume normal metabolic functions and cell division processes later when conditions improve. The core contains some interesting chemistry. It has something called dipicolinic acid in it. That's a very unique type of amino acid that bacteria use for a variety of functions. It serves in this role a protective function, helps to protect the DNA against damage. And the core also has a very high level of calcium 2+ ion content, and that both those chemicals help in maintaining a very dehydrated state. A dehydrated state ensures metabolic dormancy, because the chemistry of life requires water in order to occur. This table depicts and organizes some of the information that allows us to compare and contrast vegetative cells and endospores. So just to back up a little bit, a vegetative cell, this would be a normal, metabolically active cell. Okay? The degree of metabolic activity might vary, but the cell structure would fit, all of the information that we've been talking about in the last few days and other qualities that you've learned about cells in previous biology classes. So if you compare vegetative cells and endospores, if you look at them microscopically, vegetative cells are very non-refractile. Endospores are refractile. That means basically very reflective. And you know that because you see them reflecting light oddly when you look at them using light microscopy, typically phase contrast. The calcium content in a vegetative cell is low versus high in an endospore. As I mentioned, vegetative cells do not have dipicolinic acid and endospores do. Enzymatic activity, respiration rate, macromolecular synthesis, all of those qualities are very high in vegetative cells and very low or entirely absent in endospores. Remember, they're in a state of dormancy. And if you look at properties like heat resistance, chemical resistance, and so on, including resistance to the enzyme lysozyme, endospores are highly resistant structures, and vegetative cells are not as resistant. What's really cool, I think, is the water content. So if you look at the water content, remember, cells are generally almost entirely water. So that's true for vegetative cells, but look how little water is found in an endospore. 10 to 25% water content, and it's all entirely in the core. So these are extremely dry structures. Also, I mentioned, or I didn't mention, the role of small acid-soluble proteins. That gets abbreviated SASP, as you can see over here. These are small proteins that are made as the cell goes through the formation of an endospore. Their purpose is to complex with DNA, and they take the DNA and they convert it from one three-dimensional form to another. So they convert it from the B form that is typically found in vegetative cells to the A form. This is a much more chemically and physically resistant form of DNA, and it helps the chromosome to remain intact while it is in the endospore structure. The images on the right are taken using phase contrast microscopy to examine endospore-forming bacteria. The top image up here depicts primarily vegetative cells, and the bottom image down here shows these same cells as they are forming endospores. The endospores are the white circles. Some are still within vegetative cells, such as here, and one can be seen free-floating in the environment here. Its vegetative cell has already degraded away and just left the endospore behind. The cartoon on the left depicts the process whereby endospores are formed. This begins when the vegetative cell encounters harsh environmental conditions such as desiccation, adverse temperature, or nutrient depletion. The cell immediately replicates its chromosome. That's what's happened in here at this point. And then it partitions the two chromosomes to either side of the cell, much as occurs during routine cell division. So this is showing a partition formed. We've got a chromosome here, and we've got a chromosome here. I'm just writing DNA, but I mean an actual intact chromosome. Next, however, the cell essentially walls off one of the chromosomes and begins synthesizing the cortex and exosporium, as well as altering the chemistry of the core by synthesizing small acid-soluble proteins, dipicolinic acid, and by dehydrating the cytoplasm. Meanwhile, the vegetative cell, which is fostering and growing this endospore, is degrading around the endospore so that by the time the endospore is complete, the vegetative cell no longer exists. You can see that happening as you go around this portion of the cartoon. There's the free endospore that results from the process, and here in these steps, basically from here to here, you can see the vegetative cell degrading away as the core in here and the cortex here and the exosporium, this region, form until you get a complete free endospore. So this is a differentiation process. It's quite different from the process of normal cell division, which typically occurs by binary fission when examining most bacteria or archaea. When you have binary fission, the result is two identical daughter cells. In contrast, when you have differentiation into an endospore, you go from having one vegetative cell to one free endospore. That free endospore has all of the genetic machinery required to carry on, to live. It may be metabolically inactive, but it has the potential to become a vegetative cell again. So these endospores can last for a very long time. Many studies have shown that they can last hundreds to even thousands of years. For example, endospores have been recovered from embalmed Egyptian mummies, very ancient material, and they've been able to actually grow those organisms in the lab when they subject them to the right conditions. So the process of triggering formation of a vegetative cell, which is what would be this step here, is typically referred to as something called sporulation. It's when a spore becomes or differentiates into a vegetative cell. And the cues for sporulation are complex. We know from some organisms that a temperature spike, like a sudden increase in temperature, or an influx of water, or other environmental cues will trigger the endospore to start altering its chemistry, to become metabolically active, to interact with the environment, and to resume being a vegetative cell. This is quite relevant in the food industry. It's one of the reasons why when you have a pot of soup or chili or something else, and you take the whole pot out of the fridge, and you go from the cool temperature of the fridge to something really hot on the stove top, and then you eat a bowl, and then put the entire pot back into the fridge, you're not really supposed to do that. And one of the reasons you're not supposed to do that is that the transition from the cold temperature of the fridge to the hot temperature on the stove can serve as a temperature cue that triggers sporulation of any endospores that are within that pot of soup or pot of chili. And so you can actually increase the total cell content by driving the sporulation process, getting vegetative cells, and then those vegetative cells have the opportunity perhaps to go through regular cell division and increase the cell number to the point where your soup or your chili is either simply degrading from microbial growth or more dangerously might contain a higher number of foodborne pathogens. So that's a little bit about the life cycle of endospores. Let's consider some examples of medically relevant endospore-forming bacteria. There are 3 Clostridium species listed there: botulinum, tetani, and difficile. These are all anaerobic bacteria, and they are all endospore-forming bacteria. They all cause very dangerous disease. So Clostridium botulinum causes botulism. It's a foodborne illness. And what makes you sick is a toxin called botulinum toxin. It's in fact one of the deadliest substances known to man. Clostridium tetani causes tetanus, also known as lockjaw, and it makes a toxin called tetanus toxin. Clostridium difficile causes a disease just generally referred to as gastroenteritis. It's a particularly dangerous form of gastroenteritis and can be lethal or result in months spent in the hospital or receiving very high levels of medical care. It's a pathogen that is on the rise in the U.S. today. It's typically or often hospital acquired. So sadly, you can go into the hospital with one disease or condition, and you can leave with an infection caused by Clostridium difficile, and in fact, that may perhaps worsen your overall medical condition. Bacillus anthracis is a particularly interesting bacterium from the point of view of medical relevance and endospore-forming bacteria, because the endospores of Bacillus anthracis were the part or aspect of the organism that allowed it to be used as a successful agent of bioterror. So it's a bioterror agent. It was used in the bioterrorist attacks post 9/11 that resulted in several fatalities. Endospores were placed in ventilation systems of several buildings and in several letters, and unsuspecting individuals opened them or inhaled the air in their building. The endospores went deep into the lungs. They're very tiny, they're smaller than vegetative cells, and they went deep into the lungs, and the people didn't know they were infected, so they didn't take preventive or protective action by taking antibiotics, and most people are not routinely vaccinated for Bacillus anthracis, and so they became very quickly sick, and this is a disease. Anthrax is a disease that can escalate very quickly and cause death, particularly in cases of lung exposure, something known as pulmonary anthrax. So that is one of the reasons why all of our military troops are vaccinated against Bacillus anthracis today. Bacillus cereus is a foodborne pathogen. It causes foodborne illness, at least one particular type of Bacillus cereus, and that's where I'll leave off giving you some examples. I do want to state, however, that although I've given you examples only of medically relevant harmful pathogens that are endospore forming, the vast majority of endospore forming bacteria are simply found in a variety of environments on Earth, and they're part of degrading materials and soil and compost, and performing other normal functions that contribute to the overall, well-being and function of the ecosystems in which they occur. They're not pathogenic. With that, I'll conclude this lecture.